Hi everyone, welcome to my channel. So I'm Hello Buddy. This video I will explain about the detailed syllabus explanation about STPM Biology Semester 1. Okay, so as I mentioned, I had made the video about the introduction to STPM Biology with the syllabus, just a brief explanation and also some reference book, attention, advice, and also exam format for STPM Biology. Okay, so for those who are just graduated for SPM, you are wish to entering STPM to get biology. So this video maybe can help you to know that what you will learn in STPM Biology Semester 1. But if you have never see the video about the introduction to STPM Biology yet, maybe you can watch that video first. Because that video I will explain more detail about the introduction. So for this video, I will not explain about the exam format and attention and advice already lah. I will only focus on syllabus briefing for STPM Biology Semester 1. Okay, so as you guys, so first of all, I need to introduce myself lah. So I am the Hello Buddy. My nickname is Hello Buddy, and the full name is the Li Zixuan. So I am the current fourth year student of UTM, studying the bachelor degree of industrial biology in 2024 lah. So the video record is in 2024. So maybe some of you guys see this video, I'm graduating, yeah. So I'm actually the ex-senior STPM 2020. So already graduated almost 4 years lah. 4 years already. So my CGPA, current CGPA is at that time is 3.25. So actually I am not a very smart person or person like 4.0 for flat. So I am just a very kindful senior to help you guys as I can lah. Okay, so this one is just a repeating a reminder to you guys. So for those who already listened to that introduction to STPM Biology, maybe you already know lah. So for those who never learned yet, watch that. So you need to know that starting STPM 2026, which the students that entering from 6 in 2025, you will use the new syllabus of all subjects, not only biology but also all subjects in STPM. You will use the totally new syllabus lah. Okay, so this is just a recap lah. So as you guys know, STPM Biology along 3 semesters have 18 chapters, but for semester 1 we only have 7 chapters. Okay, so in semester 1, the theme that we need to learn is about the molecule of life and metabolism. So this is just the overall chapter that you need to learn along 3 semesters. So today we will only focus on semester 1. Okay, so so in this semester about the theme biological molecule and metabolism. So actually these two lah, biological molecule containing 4 chapters and metabolism containing 3 chapters. Okay, so biological molecule about molecule of life, introduction, molecule genetics, structure of cell organelles, membrane structure and transport. About metabolism, enzyme, cellular respiration, and also photosynthesis. Okay, so when you see the topic of this chapter, I can say that you are compulsory heard it and learn it in SPM level already. In SPM level also got this kind of chapter, right? Especially enzyme, cellular respiration, photosynthesis. Compulsory have. Compulsory you have this kind of chapter. So in semester 1 containing 7 chapters, so compared to the old syllabus, actually the difference is that add on 1 chapter, which is chapter 2. This chapter actually is coming from semester 3 originally. In old syllabus, actually is coming from here. They coming two of the topic from the old syllabus in semester 3 is transferred to the semester 1 now, becoming one new chapter. So you tell me, is it new? Actually, it's not new. It is just a transformation only. Transfer from semester 3 to semester 1. Means that you need to learn early lah. So that's why semester 1 containing 7 chapters. Add on the additional of this chapter 2. Okay. So in semester 1, I can say that almost all of the topic you already learned in SPM knowledge. So repeating again, in SPM knowledge that you learn in SPM, you need to apply it know it, the theory in STPM as well. Not say that SPM already passed in the past. Already get it, I also get A, maybe get A, whatever you get in the biology grade. But STPM, it is the new journey to you. So you should study hard lah. Not say that SPM knowledge is already past, already old. So I don't need to use it. No, in STPM, in syllabus, there are still remaining the SPM knowledge, and you need to understand and explain it. Okay. So chapter 1 is about molecules of life. So originally this chapter is biological molecule. Originally the name is called biological molecule, but in new syllabus they changed the name, molecule of life. So it's similar lah. So in molecule of life, there are 4 biological molecules that you need to learn: water, carbohydrate, lipid, and protein. So actually there are also another one biological molecule, which is nucleic acid. But nucleic acid is already transferred to chapter 2. Okay, so water, carbohydrate, lipid, protein, I think all of you already know. I compulsory SPM you already learn, but you not learn deeper. You just scan on it, screen on it, just see on it. Why? Because in SPM level, this kind of topic will never come out in essay, right? Paper 2. Normally come out in paper 1. Even in paper 2, it's just normally just one question or 2 questions. Very little. So almost all of you will not learn about this in deeper way. But in STPM, if you still remember what I said in Introduction to STPM Biology, you should, if you want to score A in STPM Biology, you should study hard and read all the topic that's mentioned in the syllabus. Okay. So what you need to learn? You need to learn about properties. Like water, you need to learn about the chemical properties, physical property of the water. And also carbohydrate, they are their property, lipid and protein also they are their property. Okay, and also the structure. What is the chemical structure of water? Water, carbohydrate, these 4 biological molecules have their own chemical structure, right? Like water, we know H2O. You need to learn what is the chemical structure and their bonding of the water. Carbohydrate also has their chemical formula, right? CHH, that one. CHH2OY, right? That one. So you need to know it. And also lipid and protein. Lipid does not have the general formula, but protein has. Protein, the monocyte is amino acid, right? So you need to know the basic structure of amino acid and also type of the amino acid. So these are all about the structure. And also classification. So this classification normally focus on protein focus on protein because protein is very complex molecule. They have their classification, also their function. And also about the polysaccharide, polymer, like polymer or carbohydrate is like starch, cellulose, glycogen, all that. Lipid including cholesterol, triglyceride, and also phospholipid, all that. So I can say that 99.99% of the content in this chapter you already learned in SPM. The difference is that you not focus on it. So this is a chance to you focus. So there is nothing new to this chapter, or totally same, totally neat, totally similar that you learn in SPM. Okay, next is about chapter 2. So this one, as I mentioned, is a new chapter and originally come from Sem 3, from the old syllabus. So this chapter is about Introduction to Molecular Genetics. Okay, so there are 3 topics that you need to learn. So one of the topic is one of the biological molecules should be in chapter 1, but they transfer into chapter 2. Okay, so this chapter 2 is about nucleic acids. Okay, so this is just very basic. You need to know about the structure of nucleotide, also the structure of DNA, RNA, and A. So I think these 3 you also already learned in SPM. What is the DNA structure and what is the bonding in DNA, and also type of RNA. We have 3 types of RNA, mRNA, RNA, mRNA, tRNA, rRNA. This one you already learned in SPM. Okay, so maybe you not deeper it. Now is the chance to let you to go into the deeper. And another one, this one is new to you, agarose gel electrophoresis. So this one is the analytical technique. Is one type of the technique in DNA analysis. You need to know the theory and the principle. Why we need to learn this? Because this one, especially when you go to degree, we want to learn about the molecular biology and also genetic. Gamma gel electroporation is the very basic technique that every time you need to do in the research to analyze the DNA. So this is very basic lah, I can say. Okay. About another two is DNA replication and gene expression. Okay. These two will be totally new to you. You never learn it in SPM level lah. Okay. So what are they? So DNA replication, if you maybe some of you know this term and some of you don't know, if you still remember that you learned in my cell division cycle about mitosis, before mitosis you have many phases, right? You have S phase, S phase, G phase, right? S1 phase, S2 phase, and G phase. And then DNA replication is happening one of the phase. I don't know already. I think it's the S phase, S2. I already forget it, but it is happening at that phase, right? But you don't need to know that phase. I just want you to make you reminding about that. So that one is the mechanism of the DNA replication. So what are they? So we know that mitosis is what? Mitosis is the copying, right? You need to make the cell. You will need the original cell and then copy to become two cells, right? So if you want to copy the cell, want to originate, want to produce the cell, I can say that simple way, produce the cell. Want to produce the cell, divide the cell. Your cell should not only have organelles, also containing the genetic material. Your genetic material must be same from the original one, from the mother cell. If not same, meaning that there are mutation and that kind will be cancer cell. But in normal situation, should be in normal way. Your genetic material should be 100% similar from the mother cell. So this is the way that how the DNA can be copied into the new cell. To copy the new DNA strain is we call as DNA replication, copying of DNA. You have one DNA, right? Now you have another cell, want to produce another cell. So that another cell need DNA. So you need to copy from the original DNA into another DNA, to another DNA. You need to copy and paste. So this copy and paste process is called as DNA replication. They have the cell have own mechanism to do it. So this is the way, the thing that you need to learn. Another one is about gene expression. Okay, so this gene expression actually is the protein synthesis. Another term, in simple term, is about protein synthesis. We know that our body actually can produce some amino acid, right? You still remember we have essential amino acid and non-essential amino acid. Only the non-essential amino acid can be produced in our body. Right? Essential is cannot produce one, if not mistaken. Okay. Non-essential is that the amino acid can be produced in our body. So how they produce? Based on this gene expression, they take the genetic material from the DNA. They take the genetic material from the DNA and then they copy. They will use the material information in the DNA with the help of the RNA to produce the protein. So there are two main processes, transcription and also translation process. So another terms that you will see in another book, they will say that it is the central dogma theory. So another terms another term, scientific terms of the gene expression will be central dogma theory. So if you're interested, you can Google it. So this two topic is totally new to you that you never learn in SPM. So normally you need to focus it. But I don't say that chapter 1 don't focus. But these two topics, I'm sure that you automatically will become focused it because it's new to you. Okay, another one is about chapter 3, Structure of Cell Organelles. Okay, so this chapter, I'm sorry, this one it should be 3, not 2. Because originally this chapter is chapter 2, not chapter 3. But now because of the addition of that chapter 2, so yeah, becoming chapter 3 lah. So this chapter, I can say that 99.99% is also, you already learned in SPM. 99.99%. This one you can see prokaryotic and eukaryotic cell. You already learned in SPM. Eukaryotic cell is like animal cell, plant cell, and prokaryotic cell, bacterial cell. You already learned, right? Cell theory, I'm sure you also already learned. And comparison of cutting and cutting style already learned. The thing that you not learn is microscope. But like microscope, I think even though you not learn about as principle in biology, but I think physics you already learn, right? The principle in the reflection topic, about the light topic in physics, you also learn, right? One of the application in microscope. So I hope that you still remember. If you already forget, never mind. Biology, STPM Biology will help you to memorize. Remember it. Okay. So this topic microscope, you will learn about the principle of the light microscope and also one thing new device we call as electron microscope to you. So you need to learn what are the difference between these two microscopes. The microscope that you see in the school, all of the school, even though in laboratory, the laboratory that you see, that you use, is we call as light microscope. So this microscope, the light microscope. So we know that microscope can be observed very microorganism, right? But light microscope have their limitation. They cannot observe too fine things. The microorganism which is very small, like virus. Virus, light microscope cannot be observed. Not even virus, like animal cell also. If you see the animal cell or plant cell under the light microscope, normally you only see the big thing one, organelles within the microscope. You cannot see ribosome one. You cannot see ribosome. Or some organelles you cannot see very detailed. You only can see the very surface only. So if you want to very see the detail, the fine structure of the organelles or even virus, you want to see the virus, you need to use the electron microscope. So you need to learn what are the difference. Why light microscope can only see the large thing, but electron can see very fine things. What? They have their principle. The answer will be their principal differences. Okay. Another one, cellular components. So you need to learn all of the organelles in animal and plant cell. So all of the organelles, so including nucleus, Golgi apparatus, chloroplast, mitochondria, cytoskeleton, vacuole, cell wall, ribosome, IER, SER, all that, almost 10, if I'm not mistaken, almost got 10 organelles you need to learn in animal and plant cell. All of them. So yeah. So in this topic also 100% you already learn in SPM, but repeatedly by compulsory know that everyone will not focus on it. You just screen only. Why? Because in SPM examination they were not coming out very detailed. But as again, I need to say that in STPM there may be come out. Because one of the year they already come out. They come out explain the function, the state 4 function of lysosome. 4 function, not only one. In SPA, you just only remember one only, or just know one function, that's all. When they not only lysosome, they will ask you explain the structure and function of the mitochondria. 8 marks. Can you explain it? Cannot, because of course, compulsory you cannot explain it in 8 marks. You only can explain it in 1 marks. Why? Because you will not focus it. You will not read down. You will not read it deeper. You only scan it, like the photostat scan only. You not very detailed in reading. Okay, so this topic very easy, is the easiest topic and also the hardest topic. Easiest is you already learned in SPM, so all of the theory you already know. Hardest part is you don't want to memorize. If you don't want to memorize, then it will become the harder part. If you memorize it, then it will become the easy part. Okay, the next topic will be the centrifugation. Okay, so this one is new to you. The new topic to you. So this one is also although new but very simple, not very hard. This one is the analytical technique to isolate the organelles from the cell. So we know that in scientific research, normally we do research about the organelles. So we only want that organelle. So for example, like mitochondria, we only want mitochondria. So how do we isolate mitochondria? So this is the technique, differential centrifugation technique, to enable us to isolate the organelles, specific organelles that we want. So this is the analytical technique. Okay, next one is about specialized cell. So this topic also 100% you already learned in SPM. So what are they? Okay, so like plant specialized cell including meristematic cell, epidermis cell, ground tissue, and also vascular tissue. All of that very familiar with it, right? All of this you already learned in SPM. So what you need to learn about, you need to learn their structure and also the function. That's all. Two things only. Similar to organelles. Organelles, you also need to learn structure and function. That's all. And plant and specialized cell, structure and function. But many, not many. Plant already got 5. Meristematic cell, meristematic tissue, epidermal tissue, ground tissue, and vascular tissue. Four. Animal got many. Animal specialized tissue containing connective tissue, muscular tissue, the nerve tissue, and also what epidermal tissue for epidermal tissue and also muscular tissue 5. So all of that you need to know. You need to know the structure and also their function. So you need to all of that you need to learn it. So this chapter 99% of their content you already learned in SPM. So very easy topic, easy chapter, but there are many things you learn. Because organelles many, right? Special cell also many. So there are many things to learn. So your school teacher possibility will not focus in this chapter because they already know you already learned it. That the difference is that you want to work hard. You want to take effort on this chapter. Most of you, I know, even me at that time, I also not take the effort. But now I already know. Because at that year, they come out the question, essay question in this chapter. And that time, I already know. If you want to score well in STPM Biology, you cannot skip any chapter. Because MPM will come out any kind of question, even though it's very easiest question like this, also they have able to come out in essay. If they come out in essay, you are very gelam. You sangat gelam. And you also very disappointed. The disappointed thing is you're not reading it. You're not read down. You're not understand it. You're not read in the deeper way. So okay, this chapter also very easy. Membrane structure and transport. So first one is about fluid mosaic model. So hope everyone know, right? All this fluid mosaic model. What is this? The structure of the plasma membrane. Phospholipid bilayer, right? And then carrier protein, all about that. And then movement of substance. So anti-transport and passive transport, you also learn it in SPM. About simple diffusion, facilitated diffusion, anti-transport, sodium potassium pump, all that. And all of that. What you learn in SPM, repeat again, learn in SPM. There are new things lah. 3 topic, new things. Endocytosis and exocytosis. So these two process is not, you cannot say it is passive transport or anti-transport. These two is the new to you. But if you remember, if you know about phagocytosis, phagocytosis is one type of the endocytosis. So you need to know about the process. And the final one is about the concept of water potential, solute potential, and pressure potential. So this is the calculation topic. And the movement of water. So normally this one in plant cell, they will give you a value about this water potential, solute, and also pressure potential, and then you need to think and then calculate it whether the water will move from cell P or cell Q, like that. Okay, so this is the chapter. Also 99% of the content you already learned, except these 3 lah. Except these 3 you not learn. The others one you also learn it. So also very easy chapter, not very hard. Okay, starting this chapter will be have many new things to you guys. Okay, enzyme. So enzyme first one is also very simple. What you learn in SPM also. Well, so this is an introduction about the enzyme. So what is enzyme? So we know that enzyme is act as a biocatalyst. In chemistry, we know you know that this is a catalyst. So what is catalyst? So they are related lah. What is catalyst? So we cap again what is catalyst and how the enzyme work. So why we need enzyme? Why we need enzyme? If no enzyme, can we survive? Or if no enzyme, what is the effect? So you need to know lah. And also another topic is the mechanism of the action. So about the lock and key hypothesis, key hypothesis, and also induced fit model, and effect of the enzyme concentration, substrate concentration, effect of temperature, and effect of the pH value of the enzymatic reaction, all that in this chapter, in this topic. So also we already learned in SPM. They are starting these three, you never learn it lah. Enzyme inhibition is the inhibitor. I don't know in SPA you heard it about or not. But I know you not learn it deeper in SPM, but in SPA you will learn it. So enzyme inhibition, there are two types of enzyme inhibition. You need to know their difference. And also, first of all, you need to know what is the enzyme inhibition. Okay, the fourth one is the classification of the enzyme. So we know that enzyme play the role in biochemical reaction, but what is the classification? So there are in the whole world, even our body system, there are many enzymes. There are thousands types of enzymes. So what are their classification? How do we classify it? So this is the classification based on the enzymatic reaction. There are scientists found that there are, if not mistaken, got seven types of the reaction in enzyme. So they classify based on that reaction. Okay, the final one is about enzyme technology. Okay, so this one is about the use of the enzyme in industry or even in scientific research or in the commercialization. So you want to use it. So normally the technique that we use is about the immobilization technique that you learn. Okay, and then according to the new syllabus, the old syllabus one, enzyme kinetic and also cofactor already removed it. So enzyme kinetic actually is the calculation of the enzyme, and the cofactor are removed. So if you refer to the senior notes or you refer to the any Malaysia publication book or Oxford Pelangi study, you see these two topics, actually you can ignore it lah. You don't need to know it lah, because it is already not in your syllabus. Okay, last two chapter, which is the cellular respiration and also photosynthesis. So cellular respiration is the chapter maybe you have heard about it in SPM. So what you learn in SPM, you also learn about aerobic respiration and also anaerobic respiration, right? Similar in STPM, you also learn this. The different thing is that in SPM, you only learn about the basic. You only learn about the equation, right? Like in SPA, your aerobic respiration equation is just one glucose molecule and then take up the oxygen molecule to produce the ATP and also carbon dioxide, right? Carbon dioxide and water. This is just the basic equation that you learn in SPM. So what you learn in STPM? In STPM is that you need to learn how our body, our cells can oxidize all the glucose molecule to become ATP. How? Why we say that one glucose molecule can be oxidized in the cell and produce 38 ATP? Why? So they have their series of the biochemical reaction. So before we learn that, you need to learn about energy in the organism. So the energy that you learn is ATP, right? So what is ATP? Have you think about that? What is ATP? ATP is energy. The real definition is that ATP is the chemical structure. So not only ATP, actually in energy there are three. ATP is the most important one, and also another two, NADH and also FADH2. You need to know their structure and also their function that act as the energy. Okay. Next one is aerobic respiration. So aerobic respiration, we know that it is using glucose to produce ATP. So how they produce? So through these three series reaction: glycolysis, Krebs cycle, and also electron transport chain. So this is the most critical part. I think every Form 6 student will focus about that. Everyone. Because and also your school teacher only will focus on that chapter. Compared to the previous 4 chapter, except chapter 2, 1, 3, 4, normally your school teacher will be fast. But chapter 2, chapter 6, chapter 7, your school teacher will be slow. Because this is new July. Okay. So you need to know how these three process produce ATP. Because you need to learn how to calculate it. Okay. And also another one, anaerobic respiration. Okay. So anaerobic respiration that you in SPM you also learn. Learn that only glucose molecule and it will partially oxidize to form 2 ATP and also carbon dioxide. Okay. So the difference is between the aerobic respiration is that not using oxygen. So why? Why no oxygen cannot produce ADP? Why? They have the reason. Why no oxygen already cannot produce ADP? Why got oxygen can produce the TA? If no oxygen, only 2. So what are the critical reaction inside there? What are the reaction inside there? Make them one of the reaction need oxygen. One of the reaction don't need oxygen. So you need to know. Okay. Chapter 7. So photosynthesis. So this chapter also you also learn in SPM, I know, but in SPM also very simple. In chapter 7, what you learn is about photosynthesis. In SPM, what? You learn photosynthesis. Also the basic chemical equation, the basic reaction in photosynthesis. What? You need to have a carbon dioxide, you have a water, and you need to have a light. These 3 things must have. And then to put, and then go through the reaction photosynthesis to produce what? Glucose and what? Oxygen. Glucose and oxygen, right? These 2 are the products. Okay. So we in SPA we also learn about 2 reaction, right? Light reaction and also dark reaction that you learn in SPM. And also you have learned about the some part lah. You know that light reaction you require light, and then you have photolysis of water, and then you will produce energy, and this energy will be used for dark reaction. And what are the dark reaction produced? And dark reaction will be use the energy and use the carbon dioxide, and then do the series of reaction. What reaction you don't know, and then to produce glucose molecule. This one that you learn in SPM, if not mistaken. That you learn in SPM. That's all. But in STPM, you will learn how light reaction is, how they produce the energy, how photolysis of water occur, and how the light act as the energy. Because we know that photosynthesis require light as the energy source. So how the light becoming energy source? Why human cannot? Why only plant can? Why only plant can? Why human cannot? Why human cannot take the light as energy source? We need to take what energy source? We need to take food. Food is our energy source. But plant don't need. Plant already directly can using light. So what are the reasons? You know that plant containing chloroplast and also containing chlorophyll. So light dependent reaction, this one is the scientific terms, which in SPA we call as light reaction. So in STPM, you need to avoid using light reaction. You need to use light dependent reaction. So depend on light. Light dependent means it depends on light. So on the other words, if this reaction does not have light, in the night condition, that's why that plant cannot do photosynthesis. Right, we know that photosynthesis only can occur in the sunny day, got the sunny light. If no sunny light, then the plant cannot do photosynthesis. So because of this, light depends on light. If there are light, then they will do. And then there are chlorophyll pigment. We know the plant containing chloroplast and chloroplast containing chlorophyll pigment. So you need to learn about the structure of the chlorophyll pigment. So we know that chlorophyll. You always say chlorophyll. So what is chlorophyll? Have you imagined this kind of question? What is chlorophyll? So what are the chlorophyll pigments? So you need to know. And then the next one is the light reaction process, the view light reaction process. So cyclic and non-cyclic photophosphorylation. There are 2 processes. So you need to know the location. We know that chloroplast that you already learned in chapter 3 about the organelles, structure of chloroplast. So where does it happen? Light reaction happen at where? I think this one also SPA you already learned. Soma and grana, right? Soma and grana. So light reaction happen in soma or grana. Think about that or about that. Okay, so we know that light reaction will produce energy and also produce oxygen. The product oxygen is from light reaction. So they produce energy. And then we know that this energy will be used by the dark reaction. So dark reaction, the scientific term is called light independent reaction. So it independent means it does not require light. Even does not have light, this reaction still occur unless it have carbon dioxide and also the energy. If there are energy and also carbon dioxide, then this reaction will directly can occur. It does not require any light one. So this one is light independent reaction. So dark reaction. So as I mentioned, you need to avoid the terms dark reaction but use in light independent reaction. So the main reaction is Karbin cycle. So they will use the carbon dioxide and the energy from the light independent reaction and through up the Krebs cycle to produce glucose molecule. Okay, so apart from that, you also need to learn about photorespiration. So there are the exceptions. Some of the plant in very hot sunny day, actually they cannot do the dark reaction. They will have the limitation cause them they do respiration. Whether they go through the dark reaction, but they do respiration. So what are the causes and what are the effects? So this one means that there is not photosynthesis. Become photorespiration. Photorespiration means what? Photorespiration means they produce the energy from light, but this energy used for respiration. So very critical, right? So you need to know that. And also, the three. So you want to avoid, if the plant want to avoid the photorespiration, there are the plants, certain plants we call as C4 plants. The leaf of this plant is very special and unique, so that they can avoid this kind of problem. So this one we call as Kranz anatomy. So before you learn this, you need to know about the basic structure of the leaf that you learn in SPM. The structure of the leaf that you learn in SPM we call as C3 plants. So C3 plant is the normal plant. Normal plant, they undergo photosynthesis. But if you want to avoid this kind of situation, some plant due to they survive at that kind of environment, they cannot avoid that. Avoid that kind of environment. They need to stay at that kind of environment so that they have their own structure, leaf structure, to avoid the photorespiration, so that it will continue the Krebs cycle to complete the photosynthesis. So we call as C4 plants. You need to know that. And then also, when you know it, you need to learn about the metabolism of the C4, also CAM. So CAM plant is another type of the plant, but their leaf structure is similar to C3, but they have their own unique metabolism process, different from the C3. This one you need to know that. So there are total 3 types of plant: C3, C4, and CAM. You need to know their difference and also learn about their metabolism. C3 is the basic that you learn directly. C3, C4, and CAM is the unique. Also, not only you learn about the theory, but also you need to know about the example. All of the example you need to know. You just know, no theory, okay, or no C4, C3, C4 is like that. In exam, especially essay, they were coming out without stating this C3, C4, or CAM. They will directly use the plant type. Fine apple. So you need to know fine apple. Okay, one of the example, fine apple. Fine apple is C3, C4, or CAM. The answer is CAM. So if you know that, so you know that. You know that if they mention fine apple, so your answer should be right about metabolism of CAM. But if you don't know, you write wrong, you write C4 or C3, then totally no marks. Because already wrong theory, wrong concept. So although you already know the concept, theory, but you also need to know their example. You don't know example, you know the theory, also usefulness. Okay, so yeah, and then the last topic is about limiting factor. Okay, so this one is the factor that affect the rate of photosynthesis that you need to learn. So we know that photosynthesis requires 3 things: carbon dioxide, water, and also the sunlight, light source. So all these 3 have their limit. They not say that you just increase one of them, then you can increase the rate of photosynthesis. No, you need to have a balance. You only increase the light, but you not increase the water and also the carbon dioxide, then the photosynthesis rate also will increase until the maximum. Until reach the maximum, then the photosynthesis rate cannot increase anymore. So this one is the limiting factor that you need to learn. Okay, so that's all about all the syllabus briefing of the semester 1 STPM Biology. So hopefully this video can be useful for those just SPM graduated that you wish to enter STPM Biology semester 1. So whatever you want to enter or not enter, if you want to enter, I wish you good luck in your STPM life, not only biology but also other subjects you need to focus, and wish you to have a good result in your STPM and can get a highly merit for applying to BU, so that you can get into the government university and the course that you like. So that's all for this video. So thanks for watching and thanks still for your support. Bye.